Okay, so for our biochemistry lab, what we're going to be doing is we're going to be testing different foods and beverages to see what nutrients are present. So there are 4 different tests that we'll be running. The first one is a simple carbohydrate test called the Benedict's test, and that's going to test for simple sugars like glucose, fructose, that kind of thing. The second test we're going to run is a starch test. It's called the Lugos test, and this is going to test for the presence of starch. Third, we're going to run a test for protein called the Biarritz test. And so that's going to test whether protein is present. And lastly, a test for fats, for triglycerides, and that is going to be the Sudan 4 test. So those are our 4 tests, but before you can actually test any kind of foods, you need to know what a positive and a negative test are going to look like. So the first thing I'm going to do is I'm going to basically run what's called a standard, sort of like a positive control group, so that you can see what positive looks like and what negative looks like. So the first standard I'm going to run is I'm going to run the Benedict's test for sugar. Now that test actually requires heat. So you say I have a hot plate set up here, and I have 2 test tubes. In one of the test tubes, I have placed some sugar solution, which is actually glucose. I can show you here, glucose here that I basically mixed up with some water. And then in my other test tube is just water, just plain old water. So to both of these tubes, I'm going to add some Benedict solution. So again, my standard is sort of my positive control to show me what a positive test looks like. And my water would be my negative control to show me what a negative test looks like. This way, when I test real foods, I'm going to know if they're positive or negative. If they turn the same color as the sugar solution, then I'll know that they were positive for sugar. If they turn the same color as the water, I'm going to know they're negative for sugar. So those actually need to heat for a second. So I'm going to let those heat. While they're heating, I'm going to show you the Lugos test for starch. So again, I'm going to get 2 test tubes. In one of them, I'm just going to put some water. So here's my water. And in my second test tube, I'm going to put what I mixed up earlier today. This is some starch solution, literally potato starch here. So this is going to be my positive control, my standard for starch. Now this one does not require heat, so I'm just going to basically pour this starch solution into my other side here. All right. And then my test for this is actually Lugos solution, which is this. It's an iodine solution. So I'm going to put a little bit of Lugos in my water. So this is a negative test, you can see. And then I'm going to put some Lugos into my starch solution. And this is a positive test. Okay, so my standard, I can get a white piece of paper so you can see these results a little bit better here. Here we go. So positive test right here. This is my standard. Notice it turns a bluish black color. And then my negative test, if something does not have starch, my water just simply turns kind of an amber color. All right, so that's my Lugos test for starch. And let's go back now because my Benedict's has had enough time now to heat. Okay, so this is my water here. You see how it's still clear blue. This is my positive test for glucose, and you see how the standard, which actually has sugar in it, turns a yellow. It can actually turn anywhere between a yellow to an orange to even a deep red color. All of those would be a positive test for sugar. All right, for my third test, slide this out of the way. My third test is going to be my protein test. So to test for protein, I have some protein solution that I mixed up here. And in my other test tube, I'm literally just going to put some water again. So here's my water here. And then my indicator this time is Biarritz solution here, Biarritz. And so I'm going to put a little bit of this in each. And then you're going to see the difference between a positive and a negative test for protein. So, all right, so here's my Biarritz in my water. And here's my Biarritz. I'll probably need to put this against some white paper so you can really see it. And here's my Biarritz in my protein. Hopefully you can see the difference here. So in a protein solution, the Burette's is going to give us, this is our standard here, right here, our positive test. Notice it's purple. And our water, our negative test is just blue. All right, so that's our protein test. All right, and last but not least, I'm going to do the Sudan 4 test. So this is Sudan 4, it's a powder. This is going to test for fat. So we're going to actually use some oil. I happen to have some mineral oil here. So we're going to put some oil in one of these test tubes. I'm not going to put a lot, actually. Oil makes a big mess. All right, so there's my oil. And in my other test tube, again, my negative control here is water. So again, my standard is my positive test so that I can see the difference. And my oil is my negative test. Okay, so I've got my water, got my oil, and now I'm gonna put a little bit of Sudan 4. So actually I'm gonna set these aside for a second because this is a solid. So I'm gonna have to dip this out and actually drop some in to each of these tubes. So I don't know how well you can see this. This is a little bit of Sudan 4 in tube number 1. Okay. So that's my Sudan 4. That up. All right. So this is my Sudan 4 in oil. I don't know how well you can see that, but it actually dissolves in the oil and turns the entire solution red. But in the water, it's actually insoluble. So the difference here, again, I can put a piece of white paper to help you see it better. So the difference here, Sudan 4 in water, insoluble. It literally stays a powder. It's just floating there on the top. But in oil, it actually dissolves. This is actually now a deep pink solution where the Sudan 4 has dissolved, all right? So that's our 4 standards. So your next step is you're supposed to make some predictions. We're gonna be testing 3 food items. I have crackers, that's gonna be 1. I have apple juice, that's gonna be our second item. And our third item, I have an egg, which I need to crack, and I will do that in my next video, and I will show you the tests. We'll run all of these tests on these items, but for now, for the apple juice, the cracker, and the egg, what I want you to do is make predictions. Again, this is just totally what you think, your hypothesis. Which nutrients do you think will be present in cracker, apple juice, and egg? And there could be more than one. All right, so do you think it'll have simple sugars? Do you think it'll have starch? Do you think it'll have protein? Do you think it'll have fat? So make those predictions, and then in the next video, I'm gonna actually run all those tests on the actual foods, and you can record your results.